Hello, everyone. Good evening or good morning, depending upon where you are. I just wanted to start by thanking Wei Kong and Tom Irving for the opportunity to talk to everyone about some very, very new data that we have. So I'm just on at the beginning of this journey of participating in experiments at Argonne, and it's been a ton of fun, as everyone has said. I'm also a believer in giving you the take-home message on the first slide or with the title. So the take-home here is that I'm going to hopefully present data to you that will convince you that C protein is in fact interacting with actin. This idea has been around for a while. This slide that I have with the myosin head has been here since 2011. We proposed in 2009 that there was C protein interactions with actin. And let's see if I can get a pointer. Maybe not. Pointer options. Okay. Laser pointer. So, okay, so that that the idea that the N-terminus of myosin binding protein C interacts with actin in some way. And so as we'll go through this, this has been a kind of a controversial idea, but I think that the scale is now tipping that this happens. So that explains the title of my talk. Let's see. Okay. Oh, it's gone. I apologize, my computer was having issues. Okay, so myosin binding protein C, we've heard a lot of speakers talk about myosin binding protein C. We're all aware that it's in the C zone of each sarcomere associated with thick filaments and a series of stripes, as shown here in this electron micrograph by Pradeep Luther, which is a misspelling there. And then the cartoon with C protein in the C zones here. Myosin binding protein C, we haven't talked a whole lot about function. We've been focusing more on structure at this workshop, but C protein, especially a cardiac version of C protein, influences myosin cross-bridge cycling rates and is necessary for enhanced contractility in response to that fight or flight response in the heart. Skeletal also becomes phosphorylated, and we'll get into that later. There's aberrant post-translational modifications, as we just heard from the last speaker, phosphorylation in particular, and changes in phosphorylation occur in heart failure. And C protein is subject to proteolysis during cardiac stress, which will be relevant for the talk in terms of the N-terminal domains. And then finally, cardiac myosin binding protein C has received a lot of attention because most mutations that cause hypertrophic cardiomyopathy are actually due to mutations in myosin binding protein C. So it's received a lot of attention for that reason. But in addition to the cardiac version of myosin binding protein C, there are two additional skeletal muscle paralogs, and these are all encoded by 3 separate genes. So there's a slow skeletal, a fast skeletal, C protein, and cardiac. And in all 3 cases, the anchoring of C protein to the characteristic stripes in the thick filament are due to domains C8, 9, and 10, and we'll show you more structural evidence for that as well, that anchors C proteins to these characteristic stripes and interact with myosin tails. On the other hand, can I advance? On the other hand, at the N-terminus of the molecule, it was determined early on that the N-terminal domains, C0 through C2 or C1 through C2 in the skeletal versions, actually bind myosin S1 and myosin S2 and regulatory light chain as well. So the N-terminus seems to interact with a variety of different regions of myosin over time. What's caused a little bit of controversy over the years since its discovery soon after C protein was discovered actually, is that the N-terminus actually can also bind to actin and tropomyosin for that matter. So the question then is, how can all of these things be true at once? To complicate things even more, additional work recently by Kamperakis the group of Camprocus, has shown that the middle domains of C protein also interact with myosin S1 and S2 domains. So there's a lot of interaction domains with myosin binding protein C. And a big question has been, how can all of these happen at once? So we'll return to that structural question in a second. But in the meantime, it seems like the consensus is that myosin binding protein C has dual regulatory effects on thick and thin filaments. So one of the first things that was noticed is that C protein has inhibitory effects in that it slows myosin cross-bridge cycling kinetics, measured in this case by KTR, which we just heard about. More recently, it's become known as a stabilizer of the SRX state of myosin. These effects are regulated by phosphorylation, and these are thought to be mainly mediated through interactions with the thick filament. On the other hand, C protein also has activating effects that have been well documented. For instance, C protein can increase myofilament calcium sensitivity, maintain force at the end of systole in active hearts in mouse models. It can slow relaxation, and it can shift the position of tropomyosin on the thin filament towards its on state. So this has all been documented both functionally and structurally, and these effects are also regulated through phosphorylation, mainly with what's thought to be interactions with the thin filament. On the other hand, there is some evidence that some inhibitory effects might be mediated through the thin filament as well. For instance, C protein can compete with myosin heads for the thin filament or for binding to the thin filament. And this is thought to account for at least some decrease in ATPase activity in solution. Okay, so what's been plaguing the field though, and why I think we're also wrapped with the new structural results, is trying to understand how all these things can be occurring at once. So early on, this is from Staranoffer's 1978 paper, where it was understood that C protein actually can interact with both the S2 region and light meromyosin, that these authors proposed that C protein might be able to hold the myosin heads to the thick filament and in that way inhibit their activity. So we now know that this is closer to true than the authors could have predicted at the time, but we know that C protein now stabilizes the interacting head motif, and probably the SRX state on the thick filament is was discussed extensively yesterday. Okay, on the other hand, my cartoon drawing here again indicates that that C protein in some way interacts with actin. And the issue was for these was that that this was somewhat controversial, not only because these are the same regions in myosin binding protein C that we're supposed to be interacting with both thick filament and the thin filament, but if you think about having a linkage across onto the thin filament, the obvious objection to this was that that would create a drag to filament sliding, and especially in the heart to diastolic relaxation, filling of the heart. So this was always kind of problematic. So the question is, how do these occur, and does this happen? Are there different populations? Or are there dynamic shifts in the movement of myosin binding protein C heads in distinct population? I can cut to the chase with this, is that we don't completely know the answer to this yet, but I think we're getting closer with some really, really exciting studies that are coming out. Okay, so speaking of exciting studies, I'm going to talk about David Tamburini's from Stefan Rauser's lab, cryo-electron tomography of relaxed cardiac sarcomeres, but there are 4 other groups who have come out with really exciting structural data in preprints as well. So the point that I want to make here, and this is data that David showed yesterday, was that basically in these reconstructions, in these tomographs, that where you see yellow here is indicated myosin binding protein C and showing its interactions with actin here. And then this is also showing where the interacting heads motifs are. And then the idea that C protein, again, is going from the thick filament to the thin filament here. So this was super exciting. And I think that there's a lot of information that we are just now digesting with these interactions. So this confirms, this data also confirms the idea that certainly C8, 9, and 10 interact with the thick filament. And as shown here, C7 has some interactions as well. But then, and again, this is something David showed yesterday, but it's, I think, one of my favorite figures, is the idea that from C7 on, when you go and you calculate the angle in the position of the N-terminal domains, the middle domains and the N-terminal domains, it appears that the myosin binding protein C is going off towards the thin filament here in all different directions. And so he plots the average 90-degree angle for which C protein goes up there towards the thin filament. But and we talked about this yesterday, for his reconstructions, the N-terminus of C protein is lost in some ways. So it could not be resolved from this. It turns out that working with Witold Balkan's group, who was just mentioned, and Christina Risi, that we've spent quite a few years actually working on how the N-terminal domains of myosin binding protein C interact with actin. So we've gotten high resolution structures of different domains interacting with actin, so we can see these on the thin filament. And I'm not going to show all of this data, but highlight just one of Christina's recent articles, JMB from last year, where it shows the C1 domain of myosin binding protein C on the thin filament, as well as a structure called the trihelix bundle of the M domain. And this is a particularly important regulatory region of myosin binding protein C. So we're getting a lot of details from these different cryo-EM approaches. And this has been really quite a revolution and very exciting to be part of this. So this is just showing you the trihelix bundle again. So it's 3 helices, 1, 2, and 3. And it's helix 3 that is shown here that actually interacts with the actin monomer. And this was an exciting result for us in my lab, because using functional methods, we had found actually in 2013 that helix 3 looked like it was the predominant binding site for actin. And so this was very confirmatory, and it was confirmatory and very rewarding to kind of see this play out structurally. So the other thing that we were interested in with the trihelix bundle is that there's mutations associated with HCM here, and I've got 4 of them highlighted. And these are not supposed to interact, or these are not supposed to really affect interactions with actin per se, but these might stabilize or destabilize the trihelix bundle. And a lot of our work that we'll talk about another time actually has to do with these mutations. But what was, again, confirmatory is that we had identified a series of charged amino acids that seem to affect actin binding affinity. And these are the ones that, again, seem to be the interacting residues that affect C proteins binding to actin here. So this was really worthwhile. And this paper by Christina was kind of a tour de force, because we finally got all of the first domains, C0, C1, M, and C2, so that we could place them individually on thin filaments and make some models and hypotheses for how C protein is interacting with the thin filament. And this is just showing 2 possible arrangements that are equally probable of C0, C1, the trihelix bundle in the M domain, and C2, for instance, and predictions based on that model then about inhibition and activation of different strands of the tropomyosin. And so this was really super interesting, because C1 seems to be the domain that interacts with tropomyosin and can move tropomyosin, whereas the other domains are all interacting with the other strand of tropomyosin. The take-home message here, though, is that taking all this data and taking what we knew about where the domains are, we built a hypothetical model of how C protein might be able to transfer from the thick filament to the thin filament. And what I want to get across here, though, is during diastole at low calcium, we were predicting that actually there would be very few interactions of C protein with the thin filament, and that was based on some of our own data as well as published data in the literature showing calcium sensitivity of the N-terminus here. But we predicted that there'd be very little interaction during diastole or at low calcium, and then that we'd get probably maximal interaction at intermediate calcium, and then there would be a clash with myosin heads at very high calcium. That clash actually could dislodge myosin binding protein C and reset the cycle. So we made some predictions based on that gets at those ideas of kind of a dynamic interplay between myosin binding protein C going from the thick filament to the thin filament. But to really test this is where we actually turn to X-ray diffraction measurements, because what I've shown you are very elegant structural studies that are static and that can get at either the thick filament really well or the thin filament really well. But I think the point has been made quite beautifully over the last couple of days that X-ray diffraction is like the tool to measure these global changes, global dynamic changes that occur within the sarcomere as a whole. So because of that, we teamed up with Anthony Hessel, who you heard from this morning, and Wei Kong and Tom Irving, and went to BioCAT at Argonne National Labs. And I think we were fortunate to be some of the last people who got to use Argonne right before its dark period here. And so as all of you guys are aware then what the experiments for this are, but we were interested in taking a muscle and understanding what effect C protein has on either thin filaments or the thick filaments and global changes, especially starting at rest, as I mentioned. So this, I want to introduce two new mouse models that we have in the lab. Some of you might be familiar with our cardiac mouse model, but basically what we've done is develop lines of mice that have a TEV protease site in them, as well as they carry a cassette for either a SPY tag or what's known as a SNOOP tag. So SPY and SNOOP are SPY tag and SPY catcher are two halves of a split protein, and SNOOP tag and SNOOP catcher are two halves of a split protein. The proteins, when you bring the two split proteins back together, then they form a covalent, instantaneous covalent bond. So they're referred to as superglue by the authors who've made them. This is Mark Howarth and the University of Oxford. Highly recommend working with Mark. He will help you with anything that you would need, and he makes all of his reagents freely available to everyone. So, but what we did was we engineered in a split or a protease site, and then we engineered in a tag, a SPY tag or a SNOOP tag, in two lines of mice. One was SPY-C1 mice that hit slow myosin binding protein C, and the second is the SNOOP-C2 mice that have the mutation in the myosin binding protein C2 gene, and this encodes fast myosin binding protein C. But we're going to focus on the SNOOP-C2 mice currently for the experiments that I'm about to show you. And what we find here is that we've decided to focus on, because this is a fast, this is myosin binding protein C expressed in fast twitch fibers primarily. We isolated the psoas muscle from mice here, and off we went to argon. So the basic experimental protocol is shown here, where we took psoas muscle from the SNOOP-C2 mice, homozygous SNOOP-C2 mice, and then under relaxing conditions, we just simply took X-ray measurements at 3 different sarcomere lengths. We then cleaved with TEV protease for 20 minutes, the exact same muscle, and we repeated the same measurements, and presumably this would be in the absence of myosin binding protein C. And I think, yes, so we've got Western blots here that show you an example of the cleavage. So what we have here is, this is fast skeletal myosin binding protein C, and I think this is actin as a loading control. When we treat with TEV, we actually cleave off the C1 through C7 domains of the protein, so the whole N-terminus of the protein. And then this is a soluble piece of protein, because we can actually just rinse the muscle and the C1, C7 fragment is removed. Conversely, if we do the same experiment with an antibody, though, to slow skeletal muscle myosin binding protein C, what you see is that slow myosin binding protein C isn't affected by the TEV treatment in these mice. So they would be in the spicy ones, which is something that we're looking forward to working with. Okay, so the X-ray diffraction results, I'm super excited to show you these. I think that you guys are the first people to see anything like this. And so I'm really thrilled that Anthony has worked with my student Nick Ingalls to actually reduce the data that I'm going to show you here. But this is one of our reflection patterns from the psoas muscle here. And the first thing that I will draw your attention to is the M2 cluster. That this was one of the first things that I received screenshots of from Nick, that showing me that this M2 cluster pre and post treatment with TEV seemed to be affected by loss of myosin binding protein C. And so this is just showing the reduced data for the intensity of the M2 cluster here, which has been associated with myosin binding protein C, but it's a bit controversial whether C protein actually directly contributes to these reflections or not. And so I'm gonna leave it at that, although we're really super excited because this the technique that we're using actually should allow us to add back even more C protein for technical reasons. And so I'm looking forward to testing the idea that these reflections are due to C protein itself. Okay, so what else did we find at a first glance of the data? And I say first glance because both Nick and Anthony have been furiously trying to reduce the data and get these results out for you. So you are, like I said, some of the first people that have seen this. So this is just talking about the equatorial results now, and beautiful review article by Wei Kong and Tom with some really nice figures showing what these patterns are giving us information on. But the first thing that we were curious about is the lattice spacing of mice, or what effect that the myosin binding protein C has on lattice spacing. And what you see is this is pre in blue and post myosin binding protein C cleavage. And what we see is a small but significant expansion of the lattice structure. And this is actually consistent with what's been reported for some of the knockout models of myosin binding protein C as well. But it wasn't necessarily expected in the sense that there is some data out there to suggest that C protein could be a rigid strut that actually keeps the filament systems from collapsing even more. So here we took a look at intensity ratio for the what's commonly considered as distribution of the myosin heads, either toward or away from actin here. And what we see is, again, an increase in intensity ratios consistent with the idea that myosin heads might be moving out towards actin. And this also was reported for the knockout mice as well by Brett Colson, I believe. So far that's consistent. But what we wanted to take a look at based on what I showed you is there any evidence for maybe effects with either thick or thin filaments in particular to help us characterize that under the resting conditions, at any rate, what filament systems C protein is interacting with. So again, this is just orientation from Wei Kong's and Tom's article, providing some handy reference figures. But we've talked about these throughout the talks yesterday and today. And what we found is that actually when you remove myosin binding protein C, we get a change in the M3 reflection or the spacing of the M3 reflection, which is attributable to the position of the myosin heads, the helical array on the thick filament here. So this, again, was consistent with this idea that C protein interacts in some way with the thick filament in the heads. The M6 spacing was also elongated, which was a bit surprising of a result, but there could be, as I'm learning more and more about how changes in one structure of the thick filament can affect these different spacings and these different reflections. I'll just leave it at the point that we've noticed this change, but I can't tell you at this point what that's attributable to in particular, but it seems like a consistent finding. Okay, so now this is the part that for me actually was the most exciting. So we also took a look at thin filament reflections, in particular the actin layer lines 6 and 7 here. So what we found here is instead of an expansion, we actually got a reduction in A6 spacing consistently across the 2,4 and 2,7. And I don't have the significant figures on here, but I also believe it's significant at sarcomere length 3. And the actin spacing or the A7 spacing was also significantly affected by myosin binding protein C. So these results are consistent with C protein interacting with actin directly under resting conditions here. And so the A6 and A7 could then be used to calculate the G-actin spacing as shown here. And that was, again, showing that consistently there's a decrease in the spacing. Troponin spacing was also decreased, and that's consistent with the changes that are going on in the actin filament itself. So these results, I personally was pretty shocked by, as I showed you that we had been speculating, hypothesizing that at least under resting conditions in cardiac muscle, now admittedly this is psoas muscle, but under resting conditions that there would be very few interactions with the thin filament, or if they were, they would be weak enough that you wouldn't really notice it. But this suggests that there are strong interactions with the thin filament at rest by C protein. So as a summary for the X-ray diffraction part of this, and then wrap up shortly after, is that basically the results are consistent with the idea that C links, these myosin binding protein C links, in fact exist in relaxed muscle, and that there's global changes in sarcomere lattice spacing, in thick filament characteristics and thin filament characteristics when you remove the C1 through C7 domains, in this case of a psoas muscle. So this was drawn for cardiac. So that was the first surprise, and I'll be really interested in hearing some of your opinions of this data. As I said, nobody's seen it yet, so this is hot off the presses. But the other thing, taking a cue from Neil Cad yesterday about kind of ending where I want to end up eventually going, is that we also did isometric force measurements in permeabilized psoas muscle, and this is Nick Eagle's work in my lab. And this was consistent with what we saw for cardiac muscle. That is basically when you remove myosin binding protein C, you get this rightward shift of the tension PCA relationship. That's shown here. It's a small shift, but it's consistently seen. And we've interpreted that as an effect on the thin filament, but as was just talked about in the last talk, changes in lattice spacing, which we've now seen could also be responsible for this. On the other hand, what we have seen, and this was originally reported for the knockout mice, is that when you remove myosin binding protein C or the N-terminus, you get this large increase in KTR rates of activation, especially at mid-ranges of force. And again, this is really consistent. But the effects that I really want to show you, just because they're just so stunning and it's been so consistent, is that while we have these functional or structural approaches, including the X-ray diffraction studies, there's properties that we're seeing with loss of C protein that we're really struggling with at the moment, but are just really astounding. But we're struggling with describing them and quantifying, and then ultimately understanding how this happens. But this is 3 force recordings that are made under pre-TEV, this is post-TEV, and this is when you add C1, C7 back. This is soleus muscle, I have to say. So this is the slow skeletal, not the fast skeletal. But what we see when we cleave with, and so presumably C protein is lost here, is that we get these really dramatic contractile oscillations. And this is now in a psoas muscle, but I will show you what these look like. Oh. Maybe I need to.
You have to ditch the pointer.
Play. It was planned this morning.
You have to turn off the pointer.
Oh wait, maybe I'll do this over here. There it goes. Okay. This will be worth the wait, you guys. So I don't know if you can see it all that well. The movie is supposed to loop, but under conditions of activation, what you see normally, you would have just a isometric contraction, but in the absence of myosin binding protein C, you get these waves of contraction that occur across the muscle fiber and stay as long as you're activating with calcium. So I think actually this is shown a little bit better, and this is a cardiac movie. And what I want you to know is that there are 3 live videos going here, but only one is going to look live. And that's this middle one where we've treated with TEV and lost cardiac myosin binding protein C. And these are activations at the same level of calcium. And what you get are these spontaneous oscillatory contractions. And so now with our 3 mouse models, we've seen this in all 3 mouse models, and it's consistent and it's reliable. And it's actually one way we verify the phenotypes of our mice is because this occurs. So one of the things that we want to do or want to know from here is basically what changes within the sarcomere, what structural changes can affect these the spontaneous oscillations to the extent that they're this reliable. And so is it a matter of lattice spacing? Is it a matter of relaxation, or what is C protein doing here? So to wrap up then, I think I've touched on that C protein has both inhibitory and activating effects through interactions with both thick and thin proteins. The interactions with the thin filament have implied for years now that C links exist, at least under certain conditions. New preliminary results that I've shown you here provide evidence for the existence of C links under relaxed conditions in psoas muscle. To be determined how loss of C proteins N-terminus predisposes to these spontaneous oscillations, and how phosphorylation in other PTMs affects C links in the SRX state. So with that, I would like to thank everyone who's contributed to this project, both in my lab and especially Anthony Hessel for this was a collaboration that kind of grew out of serendipity from a meeting, and it's just been wonderful. And then Tom and Wei Kong as well. So with that, I thank you and take any questions.
Very, very nice talk, Sam. Okay, if you have questions, please unmute yourself.
Hi, Sam. Can you hear me? Hello?
I can hear
For
you.
some reason, I'm having trouble exiting my screen share.
Okay.
Okay. Okay, so now I can look at the chat. Okay, so let's see, can you guys still hear me?
Yeah.
Okay, good, thanks. Okay, Neil Cad has asked a question. If you were to guess what length of C protein is needed to restore M3? Oh, I haven't actually even thought of that, although we're planning the next experiments for what sort of mutations and what we want to add back. So I'm not sure about that. Do you have any suggestions, Neil?
No, in short.
Any other questions? Fatima, go ahead.
Sam, do you think that the changes in the thin filaments that you showed are all along the thin filament? Because after all, the C protein would only be in the region of about, 40 to 50% of a single filament.
Yeah, I'm not sure about that. That's something that actually Anthony and I have talked about, and at this point, I'm not sure. Okay. Massimo?
Can't hear you, Massimo.
Can you unmute
Oh,
yourself?
I think you're still muted.
Yeah, sorry, I had my microphone was. Do you see the changes in the actin signals in the X-ray pattern also when you are outside the overlap between thick and thin filament? I didn't get this point.
I think I touched on that. So let me go back.
Because I saw 3 micron sarcomer length. Am I
Right,
wrong?
right. Yeah, and those spacings look like, okay, I'm just looking at the data, but I don't have the statistical significance in front of me right now, but they look like they're less prominent at 3.0. So, which is what you'd be expecting. Yeah.
Okay, thanks.
Okay. And then the troponin spacing data, were you referencing the distance between 2 regulatory units of the same filament? I will refer to Anthony for that. Anthony?
Yeah, let me read that question. And when you showed the TN3 spacing data, were you referencing the distance between 2 regulatory units in the same thin filament? Yeah. So that's the second order reflection of the troponin spacing. I'm sorry, the third order reflection of the troponin spacing. So the actual distance periodicity between troponin units along the thin filament. The stretch of the thin filament can lead to the stretch of troponin also, but you can also do, an ANCOVA to kind of tease apart, is it just caused by stretch of the thin filament, or is troponin doing something special too? And in this case, troponin isn't doing something special.
That was the part that we had talked about, yeah. Okay.